Okay, so now we're going to go through all of our different kinds of amino acids and talk about them in terms of their category. So our first category are the nonpolar amino acids. These are hydrophobic, or if you remember what that means from our lipid chapters, that means they are water-fearing. So they don't like being around water, long story short. Okay, and then the R groups are typically, or R groups are going to be either hydrogen, alkyl, or aromatic. Okay, so looking at these amino acids, we'll look at this first one here. Let's do red. So, oh, that doesn't look very good. How about pink? You still see it. It's hard to see the name if I highlight it too much. So let's maybe try darker yellow. Yeah, that's good. So as you can see here, you have your name. Let's zoom in actually. You have your name, which is in this case, glycine. You have glycine here. That is the name of the amino acid. You have your 3-letter abbreviation, GLY, and then your 1-letter abbreviation, which is G. So this chart shows you all of those different abbreviations and names. Up here on the top in yellow, that is your R group. So whatever is in yellow is going to be different from that backbone. So the blue is the backbone. By backbone, I mean you have your amine and then a carbon in the middle that has your R group, and then your carboxylic acid attached to that. So all of these throughout this whole chart are going to be formatted this way. You have your yellow R group and then your blue backbone. So that blue backbone, with the exception of one amino acid, is going to be the same throughout. And then lastly, I will point out this number here is the isoelectric point. So that is the pH at which you have your neutral amino acid. Okay, so looking at this entire chart of amino acids, let's point out some interesting details about some of the different ones. So glycine, this one that I've been talking about first, is your simplest amino acid. So it only has a hydrogen group. There's not too much fancy with it going forward. And then we also have alanine, which is just a methyl group. We have valine, which is easy to remember because the R group looks like a V, valine. So it's got an isopropyl group. We have leucine, which is like valine, but it has an extra carbon in there. We have isoleucine, which is like leucine, but one of the methyl groups is moved over. And then we have down here, we have our aromatic groups. This one's called phenylalanine. So phenyl, you have that phenyl ring in there. We also have methionine, which has a sulfur in it, which is special, has that sulfur. I don't remember what I was going to say. I'm going to skip over the next one for a second. The last one is this tryptophan. It's unique because it has an indole as that heteroaromatic group that we've talked about before. I like to remember this one's W. This is a little weird, but if you're going to try and remember all of these structures, tryptophan has a symbol of W for some reason. I don't fully understand why, but if you look, you can see that there's a W in the structure. So that's how I remember it. So there's a W in the structure. Okay, going back one to our second to last one. This is the most unique amino acid, and that's because it actually comes back and circles and has a cycle in it back to the amine. It is the only one where the R group connects back to the amine. Okay, so we have our non-polar amino acids, but we also are going to have our polar amino acids. So these are going to be hydrophilic, meaning they're water loving. And then the groups, the R groups that will be on these will be things such as, so polar R groups, and it'll be things such as hydroxyl, thiol, and amides. So looking at these R groups, we have looking at these R groups, we'll have a variety of different things. We'll have, with serine, we have an alcohol here with just one carbon in between. We have threonine, which is kind of looks like valine, where you have that isopropyl, except one of those groups is going to be an alcohol. You have tyrosine, which has your aromatic group with a phenol, so an alcohol on top of that aromatic group. Cysteine, which is a amino acid with a thiol on it. So we've talked about these a little bit before when we talked about our hair and how we have a perm and we have our disulfide bond. So that will come back up again this chapter. We also have asparagine and glutamine, which are both amide based. And something you will notice, first off, glutamine, just notice that it's Q. Asparagine is N. These are a little bit weird. Y for tyrosine. Just notice that those are different when you're looking at them. And then also something that's common with the polar amino acids is that the isoelectric point is around 6 for all of these. So it's a little bit lower than your traditional like physiological pH. Okay, then we also have our charged amino acids. They are also hydrophilic. And they have charged side chains, so charged R groups. So these will be things such as carboxylates and ammoniums. Okay, so looking at these, we have aspartate, which again has a weird letter that it is D. That has a carboxylate. We have glutamate, which is the same carboxylate, just one carbon chain longer. We also have, those are the ones that are carboxylates. So maybe we'll draw a line in between these. We also have histidine. It has this funky little heteroaromatic group here, which has the 2 nitrogens in it. We also have lysine and arginine, which are over here, and they have those ammonium kinds of functional groups on them. So when we're looking at these, you notice that, oh, that was not, let's try this again. We kind of have 2 classes. We have ones that have carboxylates. We have ones that have our ammoniums. So something to notice is that our isoelectric points are going to be very different because we have charged species. So when we look at the first ones, we have acidic amino acids. They have an acid on them, so they have carboxylic acids that got deprotonated. And so these will typically have a negative charge associated with them. They have low isoelectric points. So what this means is that you need a lower pH enough to protonate that side chain. And then when we're looking on the other side at the ammonium-based charged amino acids, it'll be the opposite. So we have more of a basic amino acids. And so these will have a positive charge associated with them at our neutral pH. So the isoelectric points are much higher. As you can see, they're pHs of 9 and 10. So we need to raise the pH enough to deprotonate the ammonium. So we have a positive charge, we need to have a higher isoelectric point to be able to deprotonate that and get it to a neutral structure. Okay, so these are all of our general amino acids or common amino acids. These, you should try and keep access to these. I will post a chart of the common amino acids. It'll have this particular chart. I'll also have one that's a little bit more colorful and fun. I personally like the other one better. But you guys should print those out, have access to those whenever you're doing these kinds of problems so that you can see the structures and all of the names and abbreviations for all of these.